Hi everyone, this is Masab Umair. I'm working as a senior scientist at National Institute of Health Pakistan. I'm really grateful to MGI for giving me this opportunity, and I'm really sorry for not being able to join the conference in person. So I'll give a very brief overview of the use of metagenome next generation sequencing in surveillance of viral diseases and in outbreak response from Pakistan's point of view. So just before I start, just to give you an overview of National Institute of Health. So the National Institute of Health the idea was conceived in 1960s, and in 1967, National Health Laboratories inaugurated. So we function as an autonomous organization, and we are the national focal point for international health regulations, global health security agenda, AMR, infection prevention and control, and national lab services, biosafety, biosecurity, and then disease surveillance, emergency preparedness. We also were designated as the National Public Health Institute of the country in 2016. And just a couple of years back, NIH has gone through a reorganization through an act. So before the Reorganization Act, there were five different divisions of the institute. However, now since the reorganization, there are seven institutes, and it includes the Center for Disease Control, Health Research Institute, Health Data Center, then National Health Laboratories Institute and of Nutrition Division, then Center of Environmental and Occupational Health, and then the Vaccine Institute. So I will mainly talk about the National Health Lab, and this is where from where I belong. So under the National Health Laboratory, we have different departments, and Department of Virology is one of those. So Department of Virology is a WHO regional reference lab for polio. Then we are also a regional reference lab for measles. We are a WHO national lab for rotavirus, gastroenteritis, and then we have a national influenza center. And then we are a national reference lab for many viral diseases such as COVID-19, dengue, and CCHF, etc. So just a comparison of how whole genome sequencing or next generation sequencing has been done over the period of last 2 decades in Pakistan. If you see before the COVID-19, around more than like 2,000 whole genomes were available for viruses from Pakistan. But just during the last 3 and 3.5 years, you can see that the number of sequences deposited on global data banks has increased quite a lot. So, but previously, most of the samples they were sent out of the country for whole genome sequence of virus, but now we have well-established next generation sequencing facilities at NIH and then in some of the provincial public health labs. So all of this uh, sequencing is now being done within the country. And among these almost 9,000 whole genomes, more than 80% of whole genomes are of SARS-CoV-2. This is just an overview of the major activities or public health interventions that were taken during the COVID-19 pandemic. So you can see that starting from COVID-19 testing in February 2020, we started genomic surveillance of SARS-CoV-2 in April uh, 2020. Then National Genomic Consortium was established at the start of 2021, and a lot of different public health intervention were taken accordingly during those different waves. So we have a National Genomic Consortium of SARS-CoV-2. It includes 13 different institutes. It has representation from both public and private sector. We have National Institute of Health leading this consortium. Then there are public health labs in the provinces that are part of this consortium. And then we also have academia that are involved in this consortium. So it's a network of 13 different institutes, and this has very actively participated in genomic surveillance of SARS-CoV-2 during the pandemic. And just to highlight here that at NIH, we basically have a well-established whole genome sequencing or next generation sequencing facility. We have the third generation and the second generation platforms. Then we also have a bioinformatics department. So we have the expertise, and this slides just shows how genomic surveillance of SARS-CoV-2 has been done during the pandemic in Pakistan. So we have reported from the initial B.1 lineage of the virus, then we have reported multiple variants. And just recently, you can see that Omicron and sublineages are being reported, and they are prevalent in Pakistan. So this slide also gives a very good indication of the next generation sequencing facility that was, or the capacity that was available at the start of the pandemic. You can see very few whole genomes are available during the first wave. However, as the pandemic unfolded, we established the NGS facility at NIH and in our provincial labs, and you can see the number of sequences have increased. Although there are very few sequences reported in the last 1 year or so, but that's mainly due to the lack of positive cases reported from Pakistan. So coming to metagenome sequencing, so Pakistan faced high, you can say, cases or a surge of conjunctivitis cases in August, September this year, where where almost like 400,000 cases were reported, and it was unknown what was the cause of this disease. So we used the metagenome next generation sequencing at NIH, and we managed to detect or identify the pathogen, and it was an enterovirus, which is the Coxsackie A24 variant. And based on our whole genome sequencing, genotype 4 was identified. And the phylogenetic analysis showed that our strain was highly similar to the virus circulating or detected in China in June 2023. So we also found some unique mutations in this virus. But again, just to highlight here the use of metagenome sequencing in identifying a pathogen that was not detectable through other assays. So we also used just recently in our province of Balochistan, there was a nosocomial outbreak of Crimean Congo hemorrhagic fever. So Pakistan is endemic for this disease, and it's a highly lethal disease. So unfortunately, we do not have at the moment primers particular for doing whole genome sequencing of this virus. So we again used the metagenome approach, and we managed to sequence 10 samples from this nosocomial outbreak of CCHF, and we found that all those samples, basically the virus, there was Asia 1 genotype across the three segments, and it belonged to clade 4A. Although the S segment was quite conserved, but we found mutations in the L and the M segment. And we also found that our strain was similar or showed high homology with strains previously reported from Pakistan as well as from regional countries, Afghanistan, Oman, India, and Iran. So again, just to highlight here, previously we used to do partial sequencing of CCHF virus based on the S segment. But you with partial sequencing, you cannot determine recombinations or mutations in other genes. So again, using this metagenome approach, we managed to sequence the whole virus, and we also found some unique mutations in the M and the L segment. So again, this highlights the importance of doing next generation sequencing or the whole genome sequencing of different viruses and pathogens. So we earlier this year we detected the first case of mpox. It was reported from a traveler returning from Saudi Arabia to Pakistan. Uh, this case was detected through PCR, and then using our metagenome sequencing approach, we managed to do the whole genome of the virus within 48 hours, and we found that the mpox virus basically belonged to genotype 8.2.1. Again, this is a very rare genotype. Most of these sequences of this genotype have been reported from India last year, so we found this genotype to be in a traveler, and then very interestingly, using the metagenomic approach, we also found that the patient was also infected with varicella zoster virus. So we retrieved the whole genome of VZV, and we found that it belonged to clade 5. So again, using metagenome approach, you can see that you can determine infection with different pathogens in a sample. Uh, just recently, we received a sample at our institute, at the virology department, of a patient who had a meningitis. Initially, he was suspected to be a case of Nipah, but we used the PCR and we found the sample to be negative for Nipah. So we again used metagenome sequencing approach, and in the blood sample we came to find out that the patient was infected with West Nile fever virus. Again, this is again something that is not common. We have a report of West Nile serological study being done in Pakistan, but the availability of sequence of the virus is not present from Pakistan. So we did that, and the virus was again showed homology with strains from India and Russia. And in the respiratory swab of this patient, we also again used this metagenome approach, and we came to identify the human respirovirus or the parainfluenza virus. So and this virus again showed some unique mutations in different segments. So again, you can see how next generation sequencing and metagenome approach has helped us in identification of the pathogens and in exploring their genomic diversity. So in Pakistan, we at NIH especially, we do surveillance of rotavirus in pediatric patients. So in children less than 5 years of age, we have a sentinel surveillance. And apart from that, we used this metagenome approach in a patient, in a child of 2 years of age, having gastroenteritis. So we came to see that the patient was infected with norovirus and sepovirus. Again, apart after rotavirus, norovirus is one of the common causes of gastroenteritis in children worldwide. So we managed to do whole genome sequencing of both those viruses. So it was a co-infection case, and we found that the norovirus genogroup 2 and genotype P15 was predominant. And so again, you can see, and then we did some phylogenetic analysis and found that it was norovirus was showing similarity with sequences from China and USA. And similarly, Cepovirus also showed homology with strains reported for globally. So again, you can see how we have used NGS quite successfully in doing whole genome sequencing of viruses and in identifying co-infection cases. So lastly, I will just conclude. We have faced certain challenges while performing next generation sequencing and especially metagenome sequencing, uh, especially with samples that showed that had high CT value of greater than 26. So it becomes quite difficult to sequence such samples. Then obviously there's a need for high coverage depth to determine virus diversity within sample. Again, this is something that is directly related with the amount of viral RNA present in the sample. And then at the national level, we have limited next generation sequencing facilities. As I mentioned, we have established NGS facility at NIH. Then in 3 of our provincial labs, we do have a NGS facility, but we still need to strengthen this, and we need to develop a few more facilities in other parts of the country where such facilities are not existing. And then sustainability is a challenge. So during the COVID-19 pandemic, we received a lot of support both from the government of Pakistan as well as from donors. But as the pandemic is slowing down, so we face this challenge of sustainability. And the way forward here is, especially for samples that have high CT value, is obviously to do culturing to increase the viral load. But again, that has some limitations of high containment, and then needing resources and reagents to do culturing. Allocation of funds, I think it's very important for surveillance, for genomic surveillance of different diseases, that funds are dedicated for such an activity. We need to invest in infrastructure, and obviously there's a need to do long-term strategic planning in order to sustain genomic surveillance of SARS-CoV-2 or other viral pathogens, because it's an expensive technology, and a lot of investments needs to be done both in infrastructure, in equipment, and then in obviously in maintaining the HR. I think this was my last slide. With this, again, I would like to thank MGI for giving me this opportunity. And if there are any questions, I have mentioned my email. You can please send me questions on my email. Thank you so much.